Greetings from Dr. K. Amala. This topic discusses the different phases of the bacterial growth curve. Bacterial growth curve. Bacteria are unicellular and prokaryotic microorganisms that most commonly replicated by binary fission through the asexual process. Bacterial growth is an increase in the total number of cells rather than the size of the cell. Microbes reproduced very fast at exponential rate under suitable condition. The exponential manner like one cell divided into 2 cells, 4 cells and 8, 16, 32 and 64 and so on. When bacteria growing on the medium, the predictable pattern of growth that can be graphically represented as the number of living cells over the period of growth totally called bacterial growth curve. The time taken for the bacterial cells to be double in their number that is called generation time of bacteria. The generation time of bacteria that is varies and depends on species of bacteria, nutritional content of that medium and environmental conditions. For example, Clostridium perfringens, it is the fast growing bacteria. So it takes 10 minutes to reach that doubling, and E. coli, Escherichia coli, the generation time that is 20 minutes, and the slowest growing bacteria, Mycobacterium tuberculosis, it takes 12 to 16 hours to reach the generation time. This is the graphical representation of bacterial growth curve. The bacterial growth curve consists of 4 different phases. First one lag phase, second one exponential or log phase, third one stationary phase, fourth one death or decline phase. In lag phase, bacteria are preparing their cell machinery for their growth and helps to adapt to grow in their new environment. And second one exponential phase. This phase characterized by cell doubling in their number. The number of new cell producing time is proportional to the present population of bacterial cells. And third one stationary phase, cells stop their growing and turn off the growth mechanisms. And fourth one, death phase, cells begin to die at an exponential rate. And this is the first phase of bacterial growth curve. When the bacteria inoculated are introduced or placed into the new environment such as new media or new broth, which provide all the essential nutrients for the growth of bacteria, but the bacteria don't start reproduction immediately after inoculation. Instead of reproduction, the first phase of new cells are metabolically active and increase in their cell size and synthesis some enzymes, factors and cofactors which is needed for the cell division under new environmental condition. In this phase, the bacteria tends to adapt itself to the new growth condition. Next to lag phase, lag is the second phase of bacterial growth curve. In this phase, cells starts dividing regularly by the process of binary fission. If any toxic substance were accumulated, it leads to stop the lag phase or slower the growth rate of bacterial reproduction. Researchers prefer in this stage of bacteria for experimental purposes. The lag phase is otherwise called as exponential phase, and in this phase marked by the doubling of bacterial cells and increases in their number. In log phase continues until there is the depletion of nutrients in the medium. The cells are healthiest at this stage of bacteria. The plotting these phases on bacterial growth curve, it gives you a straight line. And in this phase, so it can produce some primary metabolites like alcohol, ethanol, lactic acid, and certain amino acids, nucleotides, organic acids, vitamins, enzymes, and polyols. And stationary phase, third phase, the log phase begin to decline as the available nutrients become depleted and waste products start to accumulate. So depletion of the essential nutrients and formation of some inhibitory substances such as organic acid production, so the bacterial population was decreased. So bacterial cell growth reaches stationary phase where the number of dividing cells are equal to the number of dying cells. So there is no overall population growth was occur. So under the less favorable condition, competition of microorganisms for their nutrient uptake and increases and the cells become less metabolically active. And spore-forming bacteria produce endospore in this phase, and pathogenic bacteria begin to generate some toxic substance that help them to survive under the harsh condition and consequently cause diseases. And stationary phase, it can produce some secondary metabolites like antibiotic, pigments, ergot, alkaloids, naphthalene, nucleosides, peptide, quinolines, and terpenoids. And death or decline phase. It is the last phase of bacterial growth curve. In this stage, the rate of dead cells is greater than the rate of production of new cells. So all the cells that start to death due to the lagging of nutrients and physical conditions. In this death phase, the number of living cells decreases exponentially. The population of cells are declined as dying cells that can release some nutrients into the environment. These nutrients are uptaken by other microorganisms for the nutrient availability. Some microorganisms are growing. So it is helps to produce spore producing bacteria to survive long enough for spore production. The spores are able to survive under harsh environment condition of the death phase. Some MCQ questions on bacterial growth curve. In log phase, the growth is slow growth, fast growth, intermediate growth, no growth at all. The answer is slow growth. The generation time for E. coli is 20 minutes, 10 minutes, 35 minutes, 5 minutes. The answer is 20 minutes. Bacterial growth is unidirectional backward, unidirectional forward, reversible, none of the above. The answer unidirectional forward. The portion of the growth curve where rapid growth of the bacteria is observed is known as log phase, stationary phase, decline phase. The answer is log phase. In which of the following phase secondary metabolites are produced? Growth. First one log phase, stationary phase, death phase. The answer is stationary phase. Which of the following is used to grow bacterial culture continuously? Chemostat, hemostat, Coulter counter, and turbidostat. The answer is chemostat. The average size of the cell in the exponential phase is larger than the initial size, smaller than the initial size, equal to the initial size, none of these. Answer smaller than the initial size. During which phase would penicillin, an antibiotic that inhibit cell wall synthesis, be most effective? Death phase, log phase, stationary phase. The answer is log phase.